Good morning. This is Dr. Dan Guerra from VerEvMed Studios. I am coming to you from the Inland Pacific Northwest. And what we're going to do today is follow our second in a series of a discussion of bioenergetics and how we can look at basic biochemistry, that is the production of energy in cells, that aspect of it, bioenergetics, and uncover and discover some very interesting new research associated with various kinds of diseases. Now, the disease we're going to be eventually landing upon is one that is of critical nature to most in the West, and that is Alzheimer's disease. We've talked about Alzheimer's before in this series podcast from VerEvMed, and you can go back and you can look at those catalog of video lectures on YouTube by going to my Facebook page. Facebook page is down here. Now let's go ahead and create this whole thing the way it ought to be. So slideshow from the beginning. It's down here. That's my Facebook page. You can find all my video lectures there. I think there's over 45 of them now. That's me, Dr. Daniel J. Guerra, and that is my email address in blue, just below my picture. You can contact me there about any of the things we discuss here on these video lectures. And also, of course, you can contact me through my company, VerEvMed, and I'll show you all the pertinent sites for that when we get to the end of this lecture. But VerEvMed is the company that I work with, and it's the one I co-founded. And what we do is we go into the primary scientific literature, only the published literature that's been refereed that we scientists contribute to and read, and pull out the evidence from that literature, vet it in a way that is verified against the backdrop of other literature in that area, that is scientific research, and then present that as a corpus of evidence and information that can be digested and used by the people who are our clients. And these can vary from people who are practicing physicians to regional hospitals, to lawyers who need medical information, expert medical information before they go to trial, before they prepare briefs. Also for graduate students, medical students, and a large group of our clientele are also just regular folks who want to know what their diagnosis means, what their chemotherapy might mean, what their pharmacotherapy might mean, relative to the discussions with their doctor. And so we provide that kind of service, and we're all online, and you can come to our website and you can become our client. These are free lectures I'm giving. And as I said, this one particular today is part 2 of a series of lectures I'm getting into for bioenergetics. So if you want to go back and look at my previous one, it was earlier this month. So let's get started. This one is going to be specifically looking at the tricarboxylic acid cycle and the electron transport chain. And we got to get up to where we are today, which is right here. We're going to be looking at brain bioenergetics and the Sphingolipid paradigm in Alzheimer's disease. That's ultimately where we're going to get to. And if you, again, I've already discussed sphingolipids in previous lectures. You can go back and look at ones that were entirely devoted to that subject. But you don't need to do that to understand what I'm going to talk about today. I'm going to get it down to the level that this is one complete organized lecture where you're going to get all the information you need to understand what the scientific literature says about Alzheimer's disease and bioenergetics, and about how this particular kind of lipid structure, a membrane lipid structure and also signaling lipid, the sphingolipid classes, are essential to understanding Alzheimer's disease. All right, let's get started. So last time I presented this slide to you, and I'm doing it again because it's part 2. Lipid signaling controls ATP transport. We went through bioenergetics, the basics, last week. We're going to continue that a little bit with talking about the electron transport chain, but we're going to quickly get into a particular paper in the literature that's going to involve all of these topics. And that means we're going to lead into Sphingolipid catabolism and metabolism, including ceramide, mediated organelle fusion and turnover. Those later topics will be covered in later lectures. All right. Now, this is a complex slide, but I want to put everything on it for those of you that like that kind of busy slide, because this is, of course, a recorded lecture. And because it's recorded, you can go back and listen to it multiple times, or you can stop it and you can read this. So I don't feel too guilty about making it a busy slide, but I even do this in normal open lecture in classroom. So it is sort of my way of doing things. All right, this is the TCA cycle. I just want you to notice pyruvic acid right here. I like this slide because it's from a textbook I've used often in a biochemistry lecture, because it shows the structures. I think you need to understand the chemical structures to understand how metabolism works. Of course, you won't be tested on this unless, of course, you want to. Then you can contact me, and I can, of course, prepare examinations for you as you prepare for, say, an MCAT or for an upcoming biochem exam. But we're not doing that today. All right. Now, what I want you to see is that pyruvic acid can come from multiple sources. For example, lactate in the blood can be converted to pyruvate. Alanine can be converted to pyruvate. The amino acid can be converted to pyruvate. And, of course, it can come from the canonical glycolytic pathway. At any rate, you start with pyruvic acid. You go through this enzyme we're going to hover on for a while, the pyruvate dehydrogenase. It's going to take reduced coenzyme A, oxidized NAD, and it's going to decarboxylate this pyruvic acid and make carbon dioxide, which is going to be left into the atmosphere. It's a gas. And you're going to also generate the reduced form of NAD, NADH. It's going to be critical to understanding how we get energy. That very first reaction actually is going to be one of the places that we commit the reduced NADH to synthesizing ATP by its ultimate oxidation in the electron transport chain. Anyway, the product of that reaction is acetyl-CoA. Acetyl-CoA is going to condense with oxaloacetic acid, and that's where we start the narrative over here. So you take acetyl-CoA, that structure, and you're going to condense it with this oxaloacetic acid right here, and you're going to make citric acid. So this is another name for the TCA cycle, is the citric acid cycle, or the third name is the Krebs cycle. I'm sure all of you have heard of this somewhere or other in your long and storied careers. So you get a condensation of the acetyl group with a 4-carbon carrier to get a 6-carbon tricarboxylic acid, citrate. Okay, so citrate needs to be rearranged. Okay, and that's what this next reaction is called, the conatase. I'm going through this slowly because I want you to get why we need to know intermediary metabolism via this canonical pathway description and discussion, so that you can understand bioenergetics and then understand how bioenergetics is related to disease. I'm giving you the actual on-the-ground understanding like a biochemist does, or like a really good biochemistry student would get it because they'd be taking a really good biochemistry class like mine. So citrate gets rearranged, and what that means, this aldol condensation was driven by a thioester elimination, followed by a converting a tertiary alcohol, which is what citric acid is. Tertiary because it's 1, 2, the third carbon has that OH group, and that's an alcohol group. You're going to convert that tertiary alcohol to a secondary alcohol. Okay, ultimately you're going to make isocitrate. Okay, and so when you do that, it's more easily oxidizable. Okay, it's more easily oxidizable. Now that hydroxyl group is closer to this carboxylic acid, so that makes it a secondary alcohol. Okay, that's the important thing about a conatase. So citrates rearrange and oxidize with loss of carbon dioxide. That's what happens in the next step. Okay, and so you see here this isocitrate dehydrogenase reaction. This is where you again make reduced NAD to NADH. You see, the dehydrogenase is a key to understanding the TCA cycle. Every time you go through a dehydrogenase, you're going to make a reduced form of some nucleotide. This is nicotinamide adenine dinucleotide, one of the more potent ones we have in the cell. And at the same time, you're going to decarboxylate. So not always do you reduce and decarboxylase or oxidize the substrate and decarboxylate, but often you do in the TCA cycle. So you go through this intermediate oxalosuccinate, and you release carbon dioxide. So that is now generating the 5-carbon compound, alpha-ketoglutarate. A thermodynamically alpha-ketoglutarate dehydrogenase reaction, shown here. Okay, it's irreversible because, again, you're driving it with a loss of carbon dioxide. It's a second time you lose it here. You've lost carbon dioxide up here in the pyruvate dehydrogenase. You've lost it in the isocitrate dehydrogenase and now in the alpha-KDH at that enzyme. But also notice you're making a thioester, and also notice you're making NADH. All of this is coupled, and the bioenergetics of it are such that you can make a mold equivalent of NADH at the same time making a thioester, shown right there. So how do I say it here? Thermodynamically irreversible alpha KGDH. It's much like the pyruvate dehydrogenase reaction, which we're going to look at in a moment. Goes to produce a 4-carbon CoA derivative called succinyl-CoA right there. See, there's the coenzyme A. Again, that's a thioester linkage to that carbonyl. All right, and you obviously get to release the second CO2. High energy thioester is captured using succinyl-CoA synthetase. Whenever you see the T there, not synthase, but synthetase, that usually means you're going to make a either ATP or GTP. Okay, you got to make a trisphosphorylate, triphosphorylate nucleotide. That's why the T is in there rather than removing it and just having a synthase. Okay, that's a trick to understand and to remember these things. Now we've made GTP, so the high energy thioester was captured using succinyl-CoA synthetase, the enzyme, because of the way the reaction occurs, which I'm not going through that detail with you today. We would do electron pushing, no problem doing that, but not today. You get GTP, which is energetically equivalent to ATP. The reason this is GTP and not ATP has to do with having a nucleotide co-sufficiency, that is, one complementarity in purine and pyrimidine nucleotides, so you can make RNA and also ultimately DNA. That's necessary to maintain balance in the cell. And we talk about that when we talk about nucleotide metabolism, which we're not doing today. Succinate dehydrogenase is the next reaction shown there. It has a little bit less oxidizable potential, so it doesn't make NADH. It makes flavin adenine dinucleotides, shown there. That gives you, of course, fumaric acid. Okay, so that's an erstwhile member of the electron transport chain. That's actually an enzyme called complex 2 in the electron transport chain. It's actually embedded in the inner mitochondrial membrane. Okay, and so basically you oxidize succinate to fumarate. Okay, that's fumarate right there. See it? And basically that's taking an alkane and converting it into an alkene for you chemists who I know you like to always see that and for me to mention it. Ultimately, at the full end of the citric acid cycle, if we want to talk about an end, the alkene is hard to oxidize. So water is passed over that bond, and that's an enzyme called fumarase. Okay, and now you make back to making this alcohol group here. You see that? Yep. And that's malic acid, which is also carboxylic acid, of course. Now the last dehydrogenase comes along. That's the malate dehydrogenase, or MDH, and that yields oxaloacetic acid, which is where you started when you condensed with acetyl-CoA to make citrate. Okay, so there's the entire TCA cycle all on one slide. How do you like that? Probably only took me 2 minutes. So now let's get into the frank discussion here for this morning. And it is this morning, somewhat beautiful day out here in the Northwest. So we're going to be talking about in situ sphingolipid catabolism and the bioenergetic and degenerative shift in the Alzheimer's disease central nervous system. Okay, I know that's quite a mouthful, but all of this will be covered eventually in time. Just to get back at the 30,000 feet level, actually this would be about the 12,500 foot level, just suggesting. Citric acid cycle is going to generate, as we just saw, carbon dioxide, GTP, and a bunch of electrons. Not something I showed you, but that's what does ultimately happen. Those electrons, of course, are embedded in the NADH molecule. Okay, that's reducing. You reduce by adding electrons. Of course, you can make acetyl-CoA not just by what we saw there from pyruvate dehydrogenase. You can make it from fatty acids. Glucose 2-pyruvate to acetyl-CoA is the canonical glycolytic pathway. But you can also make acetyl-CoA from amino acids. For example, alanine to pyruvate to acetyl-CoA. All right. Then there's oxidative phosphorylation, which involves an electron transport chain. We're going to briefly cover right now. And that is ultimately going to be a reduction of molecular oxygen to water. Okay, one electron step at a time. Take full molecular oxygen, O2, and convert it to 2 of those and convert it to 4 water molecules. Same time, you're setting up a proton gradient, which is 36 to 38 protons. There's a little bit of leakage, and that's necessary to allow for the membrane to function correctly, actually. The proton pumping force then ultimately lets the protons within the inner membrane mitochondrial space, the IMM, those protons that pump back through a proton pumping ATPase to generate the ultimate product, ATP. Okay? This is all happening at the membrane of the mitochondria in the inner membrane. Okay? So, again, pyruvic acid lost carbon dioxide and the electrons were driven into NADH, remember? You made acetyl-CoA and you ran the TCA cycle, just showing you again where we're at. Now, this reaction, pyruvate dehydrogenase, we're going to take a little bit of a slower look at it because I want you to see the complexity of biochemistry for those of you that have never seen it. It's so beautiful. I want to show it to you, and with all the structures, of course, but also so that you understand when drugs are used to accommodate the diminution of some disease or some symptom of a disorder, and you hear that the compound you're using is an inhibitor of an enzyme, and sometimes it's an enzyme which we cover in biochemistry class. Often it would be if you took my classes, because I teach a lot of medical biochemistry. Then you would, in pharmaceutical biochemistry, you would know then how the drug works because you would know the mode of action because you would know the target of the drug. And so that's what we're going to do today. We're going to go slowly through pyruvate dehydrogenase. This is a really cool enzyme. It's actually a polyprotein that has 3 proteins associated together. It has the protein pyruvate dehydrogenase, or enzyme 1. It has the protein dihydrolipoyl transacetylase, enzyme 2. And it has a dihydrolipoyl dehydrogenase, or enzyme 3. And each of them have a certain component cofactor, which helps each of those reactions get facilitated, break down that energy barrier. So ultimately, pyruvic acid loses CO2, and you make, right here it is, acetyl-CoA from this acetylated intermediate here. You see, and there's your second product. First product, carbon dioxide. Second product is going to be acetyl-CoA. And the third product up here, when you reoxidize this reduced FAD moiety here to this disulfide, is when you carry out that electron transfer and the reduction of NAD to NADH. So those are the products: carbon dioxide, acetyl-CoA, and NADH. So take a look at this. Pyruvic acid combines first with thiamine pyrophosphate to make a hydroxyethyl thiamine pyrophosphate. That is thiamine. That's a vitamin in the diet, a B vitamin. Then that acetyl group there, okay, or now it's a hydroxyethyl group because it has the OH, is now reacted with lipoamide. This is a cofactor bound to enzyme 2. And then lipoamide, because you can break this disulfide bond and carry out now this thioester transfer, now you've made acetyl dihydrolipoamide. And there it is right there. See? Now that is going to react directly with coenzyme A with no loss of free energy, because that's also a thioester. And you're going to transfer that acetyl directly to CoA, and now you have acetyl-CoA. There's your second, really most important, probably, equally important with NADH product of the pyruvate dehydrogenase complex. Now you've made reduced lipoamide. You see here? In order to get that reoxidized, that's what this reaction does. This reaction takes now oxidized FAD, okay, and the disulfide is there, that's called oxidized, and it's going to react with the reduced form of the lipoamide to make the oxidized form of lipoamide so it can go through another round of reaction, interacting with the hydroxyethyl TPP, so you have to regenerate all of these cofactors within the enzyme. At the same time, you need now the reduced form of the FAD, right? See here? This is the 2 SH groups here. It's the reduced form. That will be sufficient amount of reducing power to reduce the oxidized NAD to NADH, again generating the oxidized FAD, which will become reduced by the reduced form of the lipoamide, and off you are again. See? So each of these reactions are coupled all in one huge protein complex in the intermitochondrial membrane called pyruvate dehydrogenase, generating all of these products. The NADH directly for electron transport chain, acetyl-CoA, for example. One of the things we're talking about today is for the TCA cycle. The other reactions can ensue for acetyl-CoA as well. All right. I want you to understand a little bit about thiamine because we brought it up. Here's thiamine interacting with pyruvate in the reaction we just saw, pyruvate dehydrogenase. And the actual initial product is acetaldehyde, TPP. You're going to have TPP also involved in a later dehydrogenase. Remember the alpha-ketoglutarate dehydrogenase we just saw when I took you around the TCA cycle. And that's going to make succinyl-CoA. And then also thiamine pyrophosphate is going to be used in a very important reaction that later on I'm going to hit on really, really hard. And that is a series of reactions in a pathway called the pentose phosphate pathway or pentose phosphate cycle. And the pentose phosphate pathway has an enzyme called transketolase, which uses thiamine pyrophosphate for the same reason to transfer carbon, 2 carbon units. And when it does it, it's going to generate actually glyceraldehyde 3-phosphate and fructose 6-phosphate. Okay, so the important point here is there's 3 times the thiamine is used. Okay, so this is just showing you that intracellular glucose metabolism, that what can happen is in the cytoplasm, glycolysis can occur, and also the OPP, and there it is again with the transketolase. And that reaction not only is you taking the 6-carbon phosphorylated sugar, glucose 6-phosphate, but it's doing a lot of really important things. It's making nucleotides. In particular, it's going to be used to make ribose 5-phosphate. And that pentose sugar, of course, is what you find in ribo and deoxyribo nucleotides. It's also necessary for the reductive biosynthesis of lipids, for fatty acid synthesis and cholesterologenesis. Within the mitochondria, of course, you have TPP bound up in those 2 dehydrogenases we just talked about, pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase. And what that is ultimately going to give you, right, because we just saw it, ATP. It's going to allow for the synthesis of amino acids, something I'm not going to talk about today. It's also going to make neurotransmitters from the amino acids, okay? Such things as serotonin. And of course, the whole aspect of the neurotransmitters involved in synaptic transmission of action potentials. So this is the thiamine, vitamin B1, TPP deficiency. So if you have a deficiency of thiamine in the diet, you tank the synthesis of ATP, some amino acids and neurotransmitters aren't made, de novo lipid synthesis doesn't occur, and all these other things occur. A shift from aerobic to anaerobic pathway, and you get increases in the concentration of pyruvate and lactate, because those would be compounds that would normally be metabolized. So it's just giving you a little bit of nutritional aspect today, where thiamine plays a role in biochemistry. Told you, what I like to do is cover all the bases when I do my lectures. So now you know about thiamine. All right, here's a very close breathable understanding of the pyruvate dehydrogenase reaction. There is the carbanion. I'm showing you the structure now of thiamine pyrophosphate. So take a look at how this works, right? So there's the carbanion form. See this anionic form of this form of thiamine pyrophosphate right here. There's a decarboxylation of pyruvate carried about by the E1 pyruvate dehydrogenase reaction, okay? The important prosthetic group, remember, is thiamine pyrophosphate, which contains this exceptionally acidic carbon atom sandwiched between a nitrogen and a sulfur. So that makes it very acidic, so that it can pick up, you see, this entire acetyl unit, right? That's what's going on here. And that's the hydroxyethyl we've got now. So you got the hydroxyethyl TPP by using this is the thiazole ring structure, which is what that is, a thiazole ring structure, is ionized to generate that carbanion. It's the first thing that happens. That carries out a nucleophilic attack on the carbonyl of the pyruvic acid, and then ultimately you make this intermediate. So I want to show you in more direct visual contact understanding how thiamine, and the thiamine portion of thiamine that we talk about is the thiazole ring, and how it's involved in catalysis. Again, taking you down to the molecular level so that you know that all of this is well worked out in biochemistry, and our drugs, pharmaceuticals, work at that level. So now let's blow back out. Let's talk about the regulation of these reactions. Pyruvate dehydrogenase, remember, is converting pyruvate, reduced coenzyme A and NAD to those products, acetyl-CoA, CO2, and NADH, right? Now, we have feedback inhibition of this reaction. This is really important to understand how this is going to link to Alzheimer's disease. So please pay close attention. Acetyl-CoA is going to inhibit this reaction. It's a product of reaction. It's called feedback inhibition. That's what the negative sign is for. Likewise, NADH at another allosteric site, that's Greek allosteros for other solid or other site, is also going to feedback because it's a product. This reaction, carbon dioxide will not, because it's free gas and it's lost. It's respired into the atmosphere. So once again, so there's control, feedback inhibition of the enzyme, and pay attention that acetyl-CoA and NADH are going to block it. Effectively blocking the pyruvate dehydrogenase is going to block any utilization of pyruvate from glycolysis that would shut down glycolytic pathway. For example, in the neuronal circuitry of the prefrontal cortex or in the limbic system or in the hippocampus or the hypothalamus, regions and nuclei of the brain, which we know to be affected in differential order and hierarchical order in various neurodegenerative diseases such as Alzheimer's disease and Parkinson's disease. Okay. So blocking that reaction by product buildup can yield a difference in the type of fuel that's used in the brain, and that's going to be a hallmark or biomarker for the progression of that disease, particularly in females, as we will see, as they age. All right. Now, that's one way of regulation, feedback regulation. The other is just taking the pyruvate dehydrogenase, okay, which is phosphorylated, and make it active by removing the phosphate, okay? Now, there's an enzyme which takes the pyruvate dehydrogenase, right? And there's a kinase of that reaction. And there are multiple regulatory components here which are going to phosphorylate that enzyme. And I want you to understand that when you phosphorylate it, so the arrow would be going in that direction, when you phosphorylate it, you make it inactive. Okay, so this is another way of understanding. Here's your product inhibition, okay? So you understand then that NADH is an inhibitor of this part of the reaction, you see, blocks that last E3, and acetyl-CoA blocks the E2, the regeneration of the lipoamide, all right? And there's your covalent modification. Once again, you have the active form of the enzyme when you remove the phosphate, and magnesium and calcium will participate in helping that reaction. Pyruvate dehydrogenase phosphatase, which is an enzyme which works on E1 to strip off the phosphate, right, from that hydroxyl group that was linked to the enzyme, amino acid. And likewise, there's an enzyme called pyruvate dehydrogenase kinase, which works on the E1 enzyme, okay, and that's where you add the phosphate, inactivating the protein. What activates that is acetyl-CoA and NADH, okay? They activate the kinase, they inhibit E1. That's why it's in red, you see? You understand that. What inhibits that are the precursor to the reaction, pyruvate, right? So you have pyruvate, which inhibits the kinase. If you inhibit the kinase, you maintain E1 in its active state. ADP, the non-tris-phosphorylated form of ATP, right? So this is now not product inhibition, right? This is substrate activation of the enzyme because it inhibits the kinase, which would inactivate the enzyme, the PDH-E1. Right? Calcium and magnesium, of course, also are going to work on the side as they did over here. And as it turns out, so does potassium, because there's a potassium voltage-gated channel involved here, particularly the synapse. All right, so once again, drilling back out, we've got pyruvic acid and fatty acids going to acetyl-CoA. Notice I'm now talking a little bit about fatty acids. TCA cycle, we already see how it works. Now we're going to take a look at the electron transport chain and making ATP. You got to make 9 ATP from 3 NADH. Okay, and you got to make 3 NADH from those 3 dehydrogenases in the pathway. Remember what they were? Isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, and malate dehydrogenase. We also had the dehydrogenase that uses FAD, right, the succate dehydrogenase, but we're not going to talk about that one because that only generates 2 ATP, but you get 2 more from that. All right. Now, they even make things more interesting. I won't say complex because it's not really complex, and it's not complicated either. It's just knowable, and so you will know it if you study it. Pyruvate not only can be decarboxylated, it can be carboxylated. Yeah. And guess what aids in the activity of pyruvate carboxylase, making, whoa, look at that, oxaloacetic acid. Now remember, it wasn't that long ago, and I know your short-term memory is up to par or better, at least cycling at the 90% level. That oxaloacetic acid is actually that intermediate, right? That 4-carbon carboxylic acid that you condense acetyl-CoA in to make citrate in the very first reaction in the first round of the citric acid TCA Krebs cycle. You choose the name you like. Notice that the enzyme is activated by acetyl-CoA. So the product of pyruvate dehydrogenase, acetyl-CoA, which is going to end up being a substrate for the next reaction, citrate synthase, is actually an activator of the enzyme, which will convert some of the pyruvate to OAA. So pyruvate is converted both to acetyl-CoA and to OAA, and the differential effect of one over the other has to do with allosteric modifications, enzyme expression, and also whatever the cell type is, if it's a muscle cell, skeletal versus heart, for example, or if it's a brain cell. Okay, how much of that pyruvate will be converted to OAA? Because you need to regenerate it. Even the TCA cycle is shown you made OAA, remember, from malate via the Nadh Dehydrogenase reaction. You notice, too, that you would be quickly losing carbon dioxide because you're running the TCA cycle. So you're effectively burning off the acetate every time you run the pathway. So, whoa, how could that then replenish? This is one of the ways it replenishes, by the Pyruvate Carboxylase reaction. Again, it's not complicated or complex. It's just the way it is. And it's knowable. All right. Now, that reaction, I want to key in on again because it actually is a target for a lot of pharmaceutical drugs. The mitochondrial form, because there's a cytosolic form as well, of Myelin Dehydrogenase. Remember that malate was oxidized to oxaloacetic acid, shown there, beautiful structures. And the NAD gets reduced to NADH. The reaction is actually favored in the reverse, okay, meaning that OAA making malate. That's exactly what happens in the cytosol almost all the time, by the way. But it only proceeds forward in the TCA cycle if the OAA is condensed with acetyl-CoA. Now you get why that pyruvate carboxylase reaction is induced allosterically by acetyl-CoA. Acetyl-CoA induces the production of OAA, and that drives the reaction to cause the condensation, making citrate. It all works out really well, you see. All right. And then the NADH, of course, is removed, and it recycles everything because the electron transport chain will oxidize that, drive electrons and protons across the mitochondrial membrane. The electrons will feed through the ubiquinol pool, of course, and all the cytochromes, ultimately yielding the reduction of molecular oxygen to water. And then the proton pumping ATPase will use the protons from the oxidation of NADH and, of course, FADH2 to a lesser degree because that makes 2 ATP, driving it through the proton pumping F0, F1 ATPase, generating all the ATP, which is what bioenergetics is in the cell. Here is the electron transport chain. This is showing you the complexes, complex 1, 2. Remember, I told you that's succate dehydrogenase, the FAD. That is part of the electron transport chain. Glasses back on. These complexes have been well understood. We understand inhibitors of all of them. A lot of potent poisons actually inhibit this pathway in various places. It's how it was worked out in the 60s in laboratories across mostly the United States, also maybe 1 or 2 in Europe, but mostly the United States. United States biochemists, educated here, of course. Complex 1 is the NADH dehydrogenase. It has a flavin mononucleotide, has a couple of iron sulfur centers. It also has the NADH ubiquinone oxidative reductase associated with it. Complex 2, we've already looked at a little bit. That's the succate dehydrogenase with the FAD. It has an iron sulfur center and a B-type heme. It also has the succinate ubiquinone oxidative reductase because you're going to be driving all those electrons into a ubiquinone pool. Now, ubiquinone, I've mentioned before in these VerEvMed lectures. You know what that is? That's a prenyl lipid, P-R-E-N-Y-L, isoprene, prenyl. Guess where that comes from? De novo cholesterologenesis. So, in order to have sufficient amounts of ubiquinol, right, you can't inhibit de novo cholesterologenesis. Yet, that's exactly what statin drugs do. They inhibit the HMG-CoA reductase, which is the rate-limiting early reaction in cholesterologenesis, blocking the production of ubiquinone. So, well, what happens then? Well, if you take enough of statin drugs, you actually get deficiency in the ubiquinone pool. Not good, because what will happen to the electron transport chain? It will be hindered, right? Because you won't have enough ubiquinone to reduce after it's been initially reoxidized to be able to receive the electrons coming from the oxidation of NADH from complex 1 or complex 2. Not good, just so you know. Here's complex 3. This has also got a ubiquinol now, the alcohol version of quinone, which is a ketone. Cytochrome C oxidoreductase, that's a component of this complex 3, but also cytochrome BC1. So there are 2 B-type hemes. Those are iron-containing proteins with this cytochrome moiety, porphyrin moiety. There's a risky named after Risky, who discovered them, iron sulfur center, and there's a C-type heme, cytochrome C1. These are all the cytochromes are. All the electrons get delivered to cytochrome C then from complex 3, and ultimately they're transported to the ultimate cytochrome oxidase, which contains cytochrome A3 complex, 2 A-type hemes, and actually now copper rather than these are all iron. And that's the complex that's going to reduce molecular oxygen to water. And that's the terminal oxidase, the cytochrome C oxidase. Okay. This is just the electron transport chain given to you again in one component slide. I really like the slide. This comes from a Devlin biochem text. So we're going to go through this real quickly just so I don't leave you in the dust and just say, well, you understand it all. Here we go. The electron transport chain is initiated by the reaction of an intermediate in metabolic reactions with the coenzyme NAD. This is an oxidation reaction where 2 hydrogen atoms or 2 hydrogen ions and 2 electrons, that's what a hydrogen atom is, are removed from the organic metabolite. The organic metabolites are usually from the citric acid cycle, as we saw. Those are the dehydrogenase reactions. And the oxidation of fatty acids, which we have not looked at today, but I've looked at extensively in other VerEvMed lectures. And over my life, I probably looked at it 10,000 times. Anyways, details following. The reaction can be represented simply by an M for big M, any metabolite. So any reduced form of metabolite plus oxidized NAD makes reduced NADH plus now what? Oxidized metabolite plus energy plus a proton. Yep, that's how it works. 1 hydrogen is removed, but 2 electrons is a hydride ion. That's the H- you see, stipulated in the architecture of these reactions. While the other is removed, is a positive ion, a proton. Usually the metabolite is some type of alcohol, which is oxidized to a ketone. Remember those alcohols? Remember the tertiary to secondary alcohol, right? Remember the alkene getting converted to malate the alcohol? Yep, I'm sure you do because we just covered it. The purpose of the other 7 steps in the electron transport chain is threefold, passing along 2 proton ions and 2 electrons to eventually react with oxygen. Remember, that's the ultimate reduction of oxygen to water, 2 O2s to 4 waters. To conserve energy by forming 3 ATPs for NADH each, and to regenerate all those coenzymes, just like we saw in the Pyruvate Dehydrogenase, another reason why I showed you that today, to their original form as oxidizing agents, right? Yeah, that was also discussed what oxidizing agents are in the previous lecture. Go right back to it. It was only a couple weeks ago, and you get all that detail. Now, once the NADH has been made from metabolite in the citric acid cycle inside the mitochondria, it interacts with the first complex 1, known as the NADH reductase or oxidoreductase. Complex 1 contains a coenzyme called FMN, which is similar to flavin adenine dinucleotide. The sequence of events is that the NADH plus another hydrogen ion enter the enzyme complex and pass along the 2 hydrogen ions ultimately to an interspace in the mitochondria. That's between the 2 membranes. Remember, the mitochondria has 2 membranes, an inner and an outer. These hydrogen ions, acting as a pump, are utilized by the proton pumping ATPase to produce ATP for every 2 hydrogen ions produced. 3 complexes, that's complex 1, 3, and 4, act in that manner to produce 2 hydrogen ions each, to thus will produce 3 ATP for every use of the complete electron transport chain. In addition, NADH passes along 2 electrons to first the FMN, up there complex 1, then onto an iron sulfur protein, and finally to coenzyme Q, which I told you is a prenolipid, which can be inhibited. Its synthesis can be inhibited by statin drugs, chronic use. The net effect of these reactions are to regenerate coenzyme A in the oxidized form, coenzyme NAD, nicotinamide adenine dinucleotide, reduced oxidized form, and the regeneration of the reactants occurs in many of the reactions so that cycling effect occurs. The oxidized NAD is ready to react further with metabolites in the citric acid cycle, those dehydrogenase enzymes. Coenzyme Q, which also picks up an additional 2 hydrogen ions to make the reduced coenzyme QH2, is soluble in the lipid matrix of the membrane. Okay? It can therefore move through the membrane and come into contact with enzyme complex 3. That's how it works. Because that's why you need it to be a hydrophobic compound, a lipid. All right. In summary, then, the very first enzyme complex in the electron transport chain is coupled with the formation of ATP. The couple reaction can be written like this: a reduced metabolite with oxidized NADA making NADH, and of course the oxidized metabolite. ADP plus P plus inorganic phosphate, PO3 plus energy makes the energy that's going to be derived from the proton pumping ATPase, the movement of that protein in the membrane. It's going to make ATP and ultimately, of course, the reduced form of oxygen, which is water. Now, another place you make NADH is in this reaction. The beta-hydroxybutyrate, which is a ketone body made actually ultimately from fatty acid oxidation. Utilization of beta-hydroxybutyrate is going to make NADH. So whenever you use ketone bodies, such as the brain will do in Alzheimer's disease in females, preferentially in the prodromal phase of Alzheimer's disease in females, hope you're paying attention, that reaction, beta-hydroxybutyrate dehydrogenase, is really going to be key because it's going to make NADH. Now, when you're making that NADH, what do you think is going to happen in all those reactions we just looked at? Well, sure, it's going to give you NADH for electron transport chain. That's why ketone bodies can be used as a brain as a biofuel. Good deal, you make ATP. But also, all of that NADH generated by this oxidation from beta-hydroxybutyrate to acetoacetate, the alcohol to the ketone, you see, of course, is going to block all those dehydrogenase reactions. It's going to shut down glycolysis, pyruvate dehydrogenase, and all the dehydrogenases of the TCA cycle. That's going to have a huge effect in neurons and in astrocytes in the brain. Okay. This is just a schematic now of the electron transport chain. I don't need glasses for this. In the intermitochondrial membrane, there's complex 1. Those putting together the FMN and iron sulfur centers, pumping out protons from NADH. More protons are coming through the quinone pool directly from the acidification of the matrix or the intermitochondrial mitosol through the quinone pool. Of course, it's generating reduction of that quinone pool. We already talked about that. Complex 3 is being now reduced through iron sulfur centers in the cytochrome Bs, recycling back to the quinone pool to pick those electrons up to the iron sulfur center to cytochrome C1, ultimately on the outer leaflet of the intermitochondrial membrane, passing those electrons now through copper, then cytochrome AA3, electrons coming back down through complex 4, is where we are, reducing oxygen to water. Yep, happens in the mitosol or the matrix. And then, of course, more protons are pumped. So all these protons are being pumped. Say, well, whoa, what are we going to do? We're going to run them through this enzyme. This is a F1 proton pumping ATPase. It's a very complex polyprotein, and it is, again, portion of it is embedded in the membrane, and a lot of it is embedded within the mitosol. So you take ADP and ATP with this complex of these subunits, these alpha beta subunits linked together. Okay, so there's 1, 2, 3, 4, 5, 6, 7, 8, an octet of them in the F1 stalk. Well, the gamma is the stalk, but the epsilon. But this oligomeric polyprotein complex up here is what actually drives the ATP synthesis, and it's driven by this molecular motor, which is turned by the protons. It's turned by the protons. Basically, this is a titration of amino acids linked in this core motor portion of the F0 portion of ATPase, which drives the reduction oxidation, yielding the turning mechanism that allows for ATP synthesis up here through the stalk, through this octet. Beautiful molecular motor. All right. Now, let's get back to a little bit more of an understanding. What's going on here is you're getting a lot of metabolite transport. Now, the intermitochondrial membrane is like a boundary. A boundary or a barrier does not allow things to go through without some kind of controlling mechanism. Not everything can pass through. If it did, you would have no control over what's inside the mitochondria and what's outside. You have to be very controlling and very specific of what's inside organelles or what's inside cells. Otherwise, they go into equilibrium with the environment, which means they die. Okay, because the environment's a harsh inorganic world. Inorganic life means you have to have facilitated and otherwise controlled diffusion and control and mediated control of metabolites, inorganic and organic, across the intermitochondrial membrane. So here's a few. Pyruvate goes in for the pyruvate dehydrogenase, for instance. Free hydroxyl ions go out, right? Phosphate goes in to make ATP. Malate goes out. Ultimately, it could be used for gluconeogenesis. Malate can go in and citrate comes out. Citrate comes out and makes it into the cytosol. Guess what that does? It gets broken down back to OAA for gluconeogenesis, but acetyl-CoA for lipogenesis, particularly fatty acid, de novo biosynthesis, and cholesterologenesis in the cytosol, right? And these all have different names. It's a monocarboxylic acid transporter, dicarboxylic acid transporter, tricarboxylic, because citrate there. Here's a phosphate transporter alone, where protons and phosphate move together. Here's the adenine nucleotide translocase. Check this out. The finished product of the electron transport chain, the business of bioenergetics, in a very narrow myopic way, but good enough for right now, is ATP. ATP leaves the intermitochondrial space. Boom, how's it get out? ADP comes in. For what? To be phosphorylated, make more ATP. Of course. You also have all kinds of amino acid transporters and carboxylic acid transporters, which are really important to understand this whole pathway. But I talked about previously, we can't spend a lot of time hovering over it today. But there's the aspartate glutamate transporter, which moves amino acids around, which facilitates actually the movement of reducing power, as does the malate alpha ketoglutarate transporter. These are involved in gluconeogenesis and also in the utilization of cytosolic NADH that you made from the glycolytic pathway, for example. Again, this is showing you as a close-up of this. Here's phosphate moving in to the mitochondria to make ATP, and here is ATP leaving the mitochondria with ADP coming in. Okay, so it's phosphate transporter here, and here is the adenine nucleotide translocase. These are proteins embedded in the intermitochondrial membrane. All right. Now, the regular stream that we just looked at through the electron transport chain, and any dysfunction that can occur, any blockage, such as with catalytic poisons, or feedback inhibition, or lack of, let me think about it, how about ubiquinone because of excessive statin use, can yield the production of reactive oxygen, shown here. So I told you that you do a 1 electron reduction of molecular oxygen. It's exactly what happens, 1 electron at a time. Oxygen first goes to superoxide and then goes to hydrogen peroxide with the addition of 2 protons, but 1 more electron then to make water. So see, 1 electron reductions, excuse me. Now you also, in this reaction here, make a hydroxyl radical, a very potent oxidant which can destroy nucleotides like DNA and RNA, the polymers of nucleotides. It can destroy DNA or RNA, make mutations, for example. It can destroy proteins, polypeptides. It can also destroy lipids. So hydroxyl radical, very potent, nasty toxin generated in the cell. When you have the electron transport chain just functioning normally, you make some of this byproduct because it's not a perfect system. Nothing is. Nothing is perfectly equivalent. Nothing is perfect in anything that we ever look at in nature, which is necessary so that it can be plastic and change over time. You understand? Regular stream through the ETC and dysfunction slows electron flow or proton motive force, will increase ROS production. That's what these are. Hydrogen peroxide, superoxide, hydroxyl radical in particular are really potent. That's the most potent ROS, reactive oxygen species. The glucose requiring OPP, oxidative pentose phosphate shunt, we looked at, it needs that thiamine for the transketolase. Remember that? That's why I showed it to you. Smuggled it in. generates NADPH. That OPP generates nicotinamide adenine dinucleotide phosphate reduced form. And that will be the ultimate reductant to get rid of all this reactive oxygen. So if you shut down glucose uptake in an astrocyte or in a neuron in the brain, and you shift from being a glucose utilizing system in the central nervous system to, say, a fatty acid oxidizing system, what happens? You don't have glucose for the oxidative pentose phosphate pathway. If you don't run the OPP, you can't make NADPH. If you can't make NADPH, reactive oxygen builds up. Reactive oxygen builds up, you get mutations in DNA, you get destruction of RNA, both ribosomal and messenger RNA, but also even transfer RNA, all three species. But you also get protein degradation and lipid degradation, and therefore degeneration of neurons and astrocytes, induction of pro-inflammatory systems from the astrocytes via the microglia, and that is inflammatory neurodegenerative disease such as Alzheimer's and Parkinson's. Yes. Remember all that iron that was there in the intermitochondrial membrane? Well, you get the reactions like the Fenton reaction. There's another hydroxyl-anion, very deadly. Another reaction, just working with superoxide and hydrogen peroxide. Remember, we just saw those being synthesized naturally in the mitochondria because of the way that electron transport chain works, or if it is diminuted in some way. And you make more hydroxyl. So it's a very, very nasty set of reactions making reactive oxygen. You have to eliminate those, which means you have to have a boatload of NADPH, and also some things like ascorbic acid, vitamin C, tocopherol, vitamin E, in their radical forms, ascorbyl and tocopheryl, respectively. And you also need glutathione, the tripeptide glutathione. Multiple ways, and some selenium, multiple ways to remove reactive oxygen, but ultimately the ultimate reducer is NADPH. A good source of that is the oxidative pentose phosphate structure. For that, you need glucose. Now, finally, let's get to a paper. I was going to do this in the next talk, but let's plow ahead. This comes from an eBiomedicine paper, so freely available online, okay, open access. Paper was published now 4 years ago, actually 3 years ago, because this is January and that was December when it was published, so 3 years and 1 month ago. Let's take a look at what the paper is going to talk about, shall we? Now, with all the knowledge you've gained from these 2 lectures, we've covered all this bioenergetics. This paper is going to be really easy to interpret and understand. Doesn't mean that it's gold. It doesn't mean the paper is going to give you some absolutely non-controversial set of data that will be converted to evidence that will be understood as verified. It could mean that this paper is just one in a series of thousands of papers produced that month or any given year that may contribute to the corpus of information we call molecular medical sciences. It doesn't mean that it can't be refuted by future research. Always remember that. What is published can always be refuted. All right, but let's take a look at this. This particular SCION, S-C-I-O-N, okay, coming from plant anatomy there, of the moving through an understanding of biomedicine actually will bear fruit, will give us buds this spring when it finally occurs, and it gives fruit to ultimate biomedical understanding of Alzheimer's disease. That's why I pulled it out of the literature. Now, myelin degeneration. Now, myelin is a lipid coating over certain cells in the central nervous system, which are myelinated, and those include astrocytes. Okay, and peripherally they're called Schwann cells. Myelin degeneration occurs during aging and in Alzheimer's disease. It also, of course, occurs in any demyelinating enzymes, right, like multiple sclerosis. White matter degeneration is predictive of cognitive decline. White matter is beneath the gray matter cortex and is composed of bundles of axons, which of course are nerve fibers, that connect neurons in different brain regions into functional circuits we call nuclei. It appears white because it supplies electrical insulation. Myelin, which is a lipid, that's what it looks like in microscopy, and that myelin covers those axons. Now that's there to act as an insulator, like an insulated wire, so that you get more rapid signal transduction, more rapid nerve impulse, if you want to call it that. formed by non-neuronal oligodendrocytes, which wrap up about 150 layers of tightly compressed cell membrane around those axons. So the non-neuronal oligodendrocytes are the cells which help contribute to the synthesis of the myelin, which is wrapped around the axon. So you need the oligodendrocytes to generate the myelin sheath around those axon fibers so that you get the correct synthesis of the central nervous system that is myelinated, and then therefore going to be the substrate for myelin degeneration diseases like AD, MS, etc. Myelin is essential, here I'm telling you, for high-speed transmission of electrical impulses, and its damage can impair conduction, signal transduction, and conduction of the electrons, and consequently sensory, motor, and cognitive functions, and one of those being memory, right? Human brain continues to myelinate, add on new myelin, which is this lipid coating, high in omega-3 fatty acids, by the way, which are found in sphingolipids, which make up the myelin sheath. So either you have a glycerol backbone, you have a sphingosine backbone, which comes from the condensation of serine and palmitic acid with a trans double bond in it. Oh yes, we talked about this previously. I'm a lipid biochemist, so it's just one of the things you always need to remember. Human brain continues to myelinate until at least third decade of age, about your 30s. And the frontal regions of the cerebral cortex, okay, which carry out higher level executive functions, are last to become myelinated in terms of developmental biology. Now, that's really interesting, isn't it? Probably because that cerebral cortex, part of the prefrontal cortex, has to receive constant input through those decades of life, all the way until about 30. Isn't that interesting? And they do say people change. Sometimes their personality changes tremendously right around that 30 age mark. I want you to keep that in mind, you millennials. All right. Now, brain regions that myelinate late, like the frontoparietal tracts, the corpus callosum, which is associated within the midline of the brain, the uncinate fasciculus, and the superior longitudinal fasciculus, are most vulnerable to breakdown and degeneration and Alzheimer's disease. Isn't that interesting? Those regions that myelinate late are the ones that are most labile to demyelination. Makes sense, less myelin. Afferent targets of those fiber systems, like the hippocampus and the subiculum, are also high in white matter. Okay, hippocampus, there you go, memory. Myelin breakdown manifests earlier in apolipoprotein epsilon 4 carriers. That's a particular type of an apolipoprotein we find actually on high density lipoprotein, which can be targeted to the central nervous system. That's right. Here's where HDL could be carrying a very toxic protein called apolipoprotein epsilon 4. And that's actually now known to be a major risk factor for Alzheimer's disease, for familial or genetic forms, not the idiotypic forms. The major risk factor for Alzheimer's disease, of course, is just plain age. After 65, every 5 years, you double the chance of getting Alzheimer's disease. Interestingly, and I guess thank God, only about 50% of people get Alzheimer's disease, no matter how old they get. For example, my mother turns 93 early next week. She does not have any symptoms of Alzheimer's disease, neither did my father before he passed a few years ago, neither did anyone in my family that anyone could know or remember. Okay, so we happen not to be ones prone to Alzheimer's disease. Isn't that great? Because if I live that long, which who knows, I can continue to be lecturing like this. And wouldn't that make everybody really happy? Probably talk a little bit slower and more gray, maybe less hair. But other than that, the same guy. At any rate, major risk factor is age. It's more prevalent in females. That is, age as a risk factor is more prevalent in females for contracting late onset AD. Get that one more time. Age is a major contributing factor to Alzheimer's disease, but it's more so in women. Okay? More so in women. So that's why this is ultimately going to lead to a paper. In fact, this paper talks about women instead of some subsequent ones, right? Women differentially get more Alzheimer's disease as they age than men. Now, one of the reasons for that that's been argued for decades, as far as I can remember, back when I was a kid in undergraduate at the University of Illinois in Champaign-Urbana, is that men die younger. And that's definitely true. And men die of things like cancer and cardiovascular disease up until recently anyway, much more frequently than women. Women are catching up fast, though, on those major causes of death, causes of mortality and morbidity, actually. But anyways, men do still die earlier than women. So that's one argument, that women have a longer time to develop Alzheimer's disease. But that's not the only reason. There's an etiologic reason why it occurs. It seems that prodromal AD can be manifested in the female brain by a switch from the glycolytic glucose utilizing pathways for bioenergetics to the utilization of ketone bodies, either generated in situ in the central nervous system or from the bloodstream, therefore ultimately from the liver. Now, isn't that interesting? Yes, it is. Now, animal model studies have shown that a drop in mitochondrial oxygen consumption, now we know how that works to make water, electron transport chain, right? Carbon dioxide release, we know where that comes from, those decarboxylating dehydrogenases. And an increase in mitochondrial hydrogen peroxide, I showed you how that happened, right? From just a normal flow of electron transport chain, are all associated with female brain senescence, that's the aging of cells, linked AD, Alzheimer's disease. And if the cytosolic phospholipase A2, sphingomyelinase pathway is involved. Now we finally get into lipids. Gosh, I'm so glad. Now, ketones versus glucose shift in prodromal AD, okay? I want you to think about this. This is important. The central nervous system bioenergetics are compromised in AD. I'm going to tell you that full stop. It's been shown in thousands of papers. Poor glucose utilization with a concomitant elevation of ketone body preference. Beta hydroxybutyrate, you just saw it, and acetoacetate, you just saw those structures. As biofuel, because it can pass right through the blood-brain barrier, right? And they can be synthesized in the liver, as I said, as a metabolic phenotype characteristic of AD. Reduction in cerebral metabolic rate of glucose utilization, particularly in the entorhinal cortex of the hippocampus, which is a major nuclei for memory reorganization. Memory's not stored anywhere, but it's reorganized there. Part of what the nuclei of the hippocampus, particularly the entorhinal cortex, is involved in. Correlate with cognitive, something I published on, with cognitive deficits over time, and most accurately predict future cognitive decline in normal individuals, as well as conversion to mild cognitive impairment in those that do not get Alzheimer's disease. Whoa, isn't that interesting? Shifting that glucose utilization in that particular subnuclei, right? That's pretty spooky. It's also good to know. Alzheimer's disease patients use a ratio of 2-to-1 glucose to alternate fuel, while controls are at, and young controls exclusively use glucose at a ratio of 100-to-1. So you can see that there's much less glucose being used in Alzheimer's patients, and the young versus older ones, right, that get Alzheimer's disease shift early. You don't want to shift early. Okay? The shift from primarily aerobic glycolysis to the ketogenic fatty acid beta oxidation pathway to white matter degeneration is where this comes from. There's the Molecular Aspects of Medicine 2011 paper. Alzheimer's disease accompanied with a decrease in expression and activity of enzymes involved in mitochondrial bioenergetics. You just saw many of those enzymes, electron transport chain, and the proton pumping ATPase, etc., and the TCA cycle, and glucose metabolism, including pyruvate dehydrogenase and complex 4 cytochrome C oxidase. Now you know where these fit in. Okay, there you go. A decreased expression of those enzymes. In situ central nervous system ketogenesis and ceramide, now you're going to bring these sphingolipids, production, are they biomarkers for AD? Maybe. During starvation and in certain reproductive states, as in gestation in females and lactation in females, lactating women after birth, post-parturition, there are massive increases in ketone bodies derived from the liver. Women go ketogenic when they're gestating and when they're lactating. And that happens from liver metabolism of lipids across, and then ultimately the ketone bodies pass the BBB, blood-brain barrier, and they go in through that monocarboxylic acid transporter. Remember, we just looked at that. Yep. Glucose transporters and MCT1 expression can also decline with age. This is where age is related as a risk factor. And with age and with normal aging and with AD aging, leading to a compensatory response that shifts to the utilization of brain-derived sources of fatty acids to generate the ketone bodies, acetoacetate, beta-hydroxybutyrate. So not only are you going to be using the ketone bodies from the liver generation, right, or during starvation, it also occurs, but also your brain starts making ketone bodies. How does it do it? By metabolizing brain-associated sphingolipid. What's that going to cause? Demyelination. What's that going to cause? Degeneration. Get it? In the aging female CNS, myelin catabolism, initiated by mitochondrial hydrogen peroxide production, now we know where that comes from, which activates a cytosolic phospholipase A2 and the sphingomyelinase pathway. Don't worry, I'm going to show you this in a moment. Degrades myelin integrity, of course, while increasing in situ lipid droplet accumulation. Yep, because of that phospholipase, you're going to start making a lot of fatty acid. Fatty acid then metabolism via beta oxidation, and then the acetyl-CoA made from the beta oxidation of fatty acids used to make ketone bodies. Beta hydroxybutyrate and acetoacetate. And then they use those through the TCA cycle, right? Source of acetyl-CoA. Here it is. This comes from a paper published in 2015, Biomedicine. We've already looked at it. There it is, November. White matter lipids is ketogenic fuel supply in aging female brain, applications in AD. Here you have in the astrocyte, mitochondria generating hydrogen peroxide, turning on phospholipase A2, making arachidonic acid. Okay. That arachidonic acid then can be used to make, which we'll show you in a second, but basically this is a compound that can be oxidized itself and can generate inflammation, generate neuroinflammation, because products of cyclooxygenase and lipoxygenase make reactive fatty acids. These are called oxygenated fatty acids, including things like leukotrienes and prostaglandins and hydroxy and epoxy tri and tetraenoic fatty acids, all of which can induce pro-inflammatory responses in situ in the brain. You need enzymes to break those things down. But the phospholipase A2 gives you the synthesis of the substrate for all that oxygenation reaction. Likewise, you get sphingomyelinase activated because of the production of the oxygenated fatty acids, which came from phospholipase A2 mediated removal from phosphoglycerolipids in the membrane of the astrocyte, thus helping to degenerate the myelin sheath. That's what that red is around the astrocyte. All that caused by mitochondrial dysfunction because of aging and normal wear and tear on the ETC, electron transport chain, because of hydrogen peroxide production. You get sphingomyelinase activation, which causes this. The myelin is broken down to ceramide because of neutral sphingomyelinase. Ceramide itself is going to be really important later in these lectures, but you already know, or I've told you in multiple lectures, ceramide is involved in apoptosis, programmed cell death, right? Really potent lipid, sphingolipid, which is involved in the induction of cellular programmed death, okay? Programmed cellular PCD. Also, there's an enzyme which takes the fatty acid off ceramide, giving you fatty acids, which can be used directly in beta oxidation in the astrocyte. And then that acetyl-CoA generated from beta oxidation will be used in ketogenesis, making ketone bodies. They'll leave the astrocyte via the minocarboxylic acid transporter. Remember that? Leave it using the same transporter now in the membrane of the neuron, bringing the ketone bodies in, using ketone bodies preferentially rather than glucose. Now, ketone body utilization, regenerating acetyl-CoA with the inhibition of glucose utilization in the neurons, right? Because that's what's going to happen, okay? All right. So you make ketone bodies in the astrocytes too, which can be sent directly there, or you can synthesize them in situ. So you have it happening in the astrocyte and neuron, and this coupling together is going to degrade white matter. It's going to degrade the sphingomyelin to a highly potent not only pro-inflammatory fatty acid metabolites, but also pro-apoptotic metabolites like ceramide, and also feeding some of those fatty acids also directly ketogenesis and utilization with blockage of glucose, all of which are prodromal and also found in later stages of full-blown Alzheimer's disease. And all of this occurs more frequently in females than men. Okay. So here's just a schematic from one of the previous lectures. This comes from the Journal of Cell Science, a 2005 paper from Sarah Spiegel's lab. Okay, so basically just take a look at this. Start off with sphingomyelin. Sphingomyelin is broken down by sphingomyelinase to make ceramide. Ceramide generates apoptosis. The rest of cell cycle, no more DNA synthesis and RNA synthesis, and cellular senescence, so aging and programmed death. Ceramide can be worked upon by phosphorylation. There's a ceramide kinase making ceramide 1-phosphate. That actually works as cell survival cofactor. It proliferates cells, and it also induces inflammation. You say, well, cell survival, good. You don't want neurodegeneration. So proliferation, good, maybe. What about inflammation? If you get neuroinflammation and cell proliferation, then you can get what? Brain cancer, right? Megaloblastomas. So this turning on this pathway is also not good. You don't want any of this happening. Now there's an enzyme actually which will dephosphorylate it, putting ceramide back in the pool and causing apoptosis. Likewise can be used to resynthesize sphingomyelin if sphingomyelin synthase is actually functioning there. Here's the de novo synthesis of ceramide. So you can make from the de novo synthesis ceramide synthase right there. You can make it from dephosphorylating ceramide 1-phosphate. You can make it from sphingomyelinase activity. So you have a lot of ways to make ceramide. Likewise, you can start from sphingosine. Ceramide synthase is going to make ceramide. There's an enzyme called ceraminidase, which produces sphingosine, and there's a whole other signaling pathway here. Sphingosine also induces apoptosis, like ceramide, and also causes cell cycle arrest. It can be phosphorylated to sphingosine 1-phosphate, which does a couple of different things. It causes cell motility, different from the ceramide, phosphorylated ceramide, but also induces cell survival, cell proliferation, inflammation, all of which can be dangerous in the brain. You can also break down sphingosine 1-phosphate to ethanolamine 1-phosphate and palmitaldehyde, and these ultimately can be also used as precursors for other lipid pathways. So this gives you just a bioactive sphingolipid metabolic route. You see how many of these are interconvertible, and because of that, you'll make a lot of potent lipids, and all of this is induced, remember, because lack of glucose utilization. They call that hypoglucose metabolism. Okay, hypo being below, right? In the brain. Prodromal Alzheimer's disease because of the utilization of fatty acids, which can turn on the production of all these sphingolipid intermediates by turning on sphingomyelinases because of, again, arachidonic acid oxygenation coming from phospholipase A2, coming from things like hydrogen peroxide and also other reactive oxygen species. All right. Now, take a look at this, putting it together with metabolic pathways. You have glucose going through glycolysis. These are hexokinase and phosphofructokinase, the pyruvate. These are gatekeepers of the glycolytic pathway we've talked about before. Pyruvate enters the mitochondria, the Pyruvate Dehydrogenase reaction. Remember, it can be taken out of reaction by phosphorylating it. What else did I tell you will knock it out? NADH and acetyl-CoA, okay, which can be coming from fatty acids now. Acetyl-CoA entering the TCA cycle. There's the alpha-ketoglutarate dehydrogenase. There's all those dehydrogenases making NADH. This is the electron transport chain. It's not showing you that succinate dehydrogenase here, but it's complexes 1, 3, and 4. You have chrome pull here, of course, and all the cytochromes building up ultimately the production of ATP. Okay? Now, at the same time, peripheral ketone bodies can be entering. Acetoacetate can be generated inside the mitochondrion. You transfer coenzyme A to it so that it can be put in metabolic play. This reaction also uses the succinyl-CoA, which is going to remove that from the TCA cycle and remove that FADH signaling as well. The acetoacetyl-CoA then gets broken down to acetyl-CoA, because you make 2 of them, because you only have 1 CoA here. And that acetyl-CoA then is going to enter the TCA cycle subsequent to the pyruvate, and it's going to generate NADH by its own method, right? Because it's running the TCA cycle. that you run through the electron transport chain. So here's the peripheral ketone body utilization switch from glucose. That's what's showing you, again, what we just looked at here. Phospholipase A2 activation is the enzyme that removes the fatty acid from the 2 position of preformed membrane-linked phosphoglycerol-containing lipids. That causes white matter degeneration, fatty acid beta oxidation, making ketone bodies, of course. Remember all of this. The carboxylic acid transporter getting the neurons from the astrocytes. We saw this. All of this is the switching. So that is all we're going to talk about today. We're going to continue on with this, and then we're going to ultimately get to a paper that was just published this month. I talked about it last segment of this, who knows, maybe 3, 4, 5-part series of talking about how bioenergetics is linked to neurodegenerative diseases like Alzheimer's disease. We're keying in on that later. We're going to bring this all together. We're also going to talk about more lipid metabolism and ultimately maybe a couple of other really important pro-inflammatory diseases of the brain and other regions of the body. Again, that's me, Dr. Dan Guerra, in black and white with my wood behind me and my chainsaw called Tangerine, cutting wood out in the Pacific Northwest. There's my email address. You can contact me directly about any content I just generated for you in this talk. There's my company, VerEvMed. There is our email to contact us directly through that site. And there's the website. Please reach out and take a look at it and contact us either via this email or my personal email. We'd be glad to have you as a client. Again, this is my Facebook page where all of these lectures are found. This picture here I did not take, although someday I want to. Hint, hint, for those that want to buy me a vacation to where? Slovenia. And this is Lake Bled. Okay, that's a Lake Bled castle, and that's the Church of the Assumption of Mary on this little island in Lake Bled. And this is also all in Slovenia. Now, why is this important to me? Because my mother is turning 93, who does not have Alzheimer's disease, is from, her family's from Slovenia. And actually, I'm half Slovenian. So it's an interesting thing that I like to look at beautiful pictures, and I love the fact that Slovenia has mountains. Obviously, they're in my blood because I love the Rocky Mountains and I live near them. So anyways, that's what that's a picture of, and I took that directly off of a website. And like I said, ultimately, I would like to spend a night in that castle. So let's shut this off. And let's do like what I always try to do at the end of our talk, although the light really built up back there, which is to say